Good afternoon, everyone. Today we are discussing the antiviral drugs pharmacology, and antiviral drugs are divided into the groups depending on the which type of the virus they are prescribed for, and so they can be divided to anti-influenza drugs, so that are acting on this influenza virus, and second group is anti-HIV, that is most of the new drugs and most diverse group, and also anti-herpes drugs. Anti-herpes drugs. This is a herpes of simplex virus, and also and anti-hepatitis drugs. So all these viral particles are uh, different in structure and the type of the RNA or DNA, and also the mechanism in which they incorporate into the cell. So that is why different groups of the drugs are prescribed for the treatment. So, and the first type of the virus is influenza virus, and influenza virus is consists of uh, of uh, add, from the RNA uh, a stand, and that is enveloped in the uh, capsid with the hemagglutinin, neuraminidase, and matrix 2 proteins that are located on the surface of the uh, uh, viral particle, and to enter the cell, the hemagglutinin will react with the sialic acid on the surface of the human cell, and that makes the engulfment and entrance to the cell. And once inside the cell, the matrix 2 ion channels will allow the acid or the positive ions enter the cell, and that acidifies the inside of the capsid, and that allows to release the genomic material to the nucleus of the cell, and then the RNA is used to transcript to the matrix RNA, and then this matrix RNA is used to produce proteins, the viral proteins in the cytosol of the cell. And then these proteins, viral proteins, including the neuraminidase, hemagglutinin, and M2 protein, are released by the Golgi apparatus to the surface of the cell, or they can go back to the nucleus and will help to replicate the RNA. So after that, the new replicated RNA is can be bought off with the parts of the membrane, with the viral proteins already incorporated into the membrane, and then the neuraminidase proteins will remove the sialic acid from bonds to allow the new particle to bud off from the cell. So that this way the new viral particles are released from the cell, and that can infect all the cells nearby. And the drugs that are used for the treatment are divided in three classes. So first class is M2 ion channels inhibitors, and this group is only effective against influenza A type, so it's not effective against influenza B because of the M2 protein uh, structure differences and mutation. So, and these drugs include amantadine and trimantadine. So, then the next group is the endonuclease inhibitors, and this is a new group that with the only member baloxavir, and baloxavir is inhibiting endonucleases that are required for the mRNA or, production in the nucleus, and this way inhibiting one of the steps in the viral multiplication. And the third one, but not the least, is the neuraminidase inhibitors, including oseltamivir, peramivir, and zanamivir. And these drugs are inhibiting the neuraminidase proteins that are required to ward off the new particles from the cell. So these are the most effective drugs for the treatment of the influenza, and the next group is HIV, and HIV is more complex virus and includes three layers of the viral particles. So the first is GP120 and GP41 proteins, and then there is proteases and integrases of proteins, and then also RNA, two strands of RNA of the viral RNA with the reverse transcriptase proteins. So these the entry begins with the interaction with the CD4 receptor at the CD4 positive T lymphocyte. So this is a CD4 positive T lymphocyte that is the most important cell in the immunity, in the human immunity, and the entrance begins with the interaction with the CD4 receptor that is located at the surface of the T lymphocytes, and this interaction is done with the help of the co-receptor CCR5 or CXCR4. And after that, the particle enters the cell, and the RNA is capsid with the RNA is released, and this capsid is used to make a double-stranded DNA from the RNA of the viral RNA, and this double-stranded RNA further is interfering with the integrase, and with the help of the integrase it can be integrated to the human DNA, and this way the human transcription and translation mechanism, the apparatus is used for the synthesis of the viral proteins, including the viral RNA, and then these proteins are covered with the capsid and bought off from the cell the same way, so as influenza virus. So and to treat the HIV uh, infection, uh, the 5 types of the groups are used. So, and first group is the entry inhibitor, and entry inhibitors include and Fulvirtide and Maraviroc and Ibalizumab. So these drugs block either CD4 receptor or CDR5 or CXCR4 receptors, thus inhibiting the entrance of the capsid to the cell nucleus, to the cell, and preventing the T lymphocyte integration. So these are the first step of the integration. So the next group is nucleotides reverse transcriptase inhibitors, and these drugs will prevent the development of the double-stranded DNA from the viral RNA, and they inhibit the reverse transcriptase that is located in the viral capsid, and further that is released with the viral RNA to the cytoplasm of the cell. And these include abacavir, emtricitabine, lamivudine, tenofovir, zidovudine, and many others. So the next group is inhibiting the next step in the viral RNA integration. It is a non-nucleotides reverse transcriptate inhibitors. They include doravirine, efavirenz, entravirine, nevirapine, and rilpivirine. So these are drugs that are also inhibiting the reverse transcriptase inhibitors, but they are not nucleotides. So the previous group was nucleotide reverse transcriptase inhibitors that are actually look like the nucleotide, and that is why they inhibit the reverse transcriptase. And the second one is a non-nucleotide reverse transcriptase inhibitors that are chemically they are not relevant to the RNA or DNA, but they inhibit the reverse transcriptase directly. So either way, the viral DNA double stranded is not formed, and that is why it cannot be used for the integration, for the further integration. And the next step that can be inhibited is the integrase inhibitors, and that include the dolutegravir and dolutegravir. And these drugs inhibit the integrase protein that integrates the double-stranded viral DNA to the double-stranded human DNA. So and that happens in the nucleus. And the last but not the least group is the protease inhibitors, and protease inhibitors will inhibit the viral proteases that will cut the proteins from the that are come off from the translation, and so they are not yet active just after the synthesis on the matrix RNA, and they require the cutting with the proteases to the active forms that will like divide each protein, so they can be used for the viral particle assembly. So and that after that the assembled particle will, the capsid will enclosed by the membrane and then burst off from the cell. So these are usually for the treatment at least 5 or even 7 drugs are combined for the treatment of the HIV because there are different types, and also to reduce the doses and to increase the efficiency, usually up to 14 different drugs can be combined for the treatment of the HIV patients depending on the severity of the disease. So and then next group is hepatitis C virus, and there are two most commonly seen hepatitis, hepatitis B and hepatitis C, that have drugs that can that are effective for the treatment. So hepatitis A, that there is no treatment available, and for the hepatitis C, the viral DNA. So the viral is one standard RNA, and it engulfs to the cell, and then the RNA is going to the Golgi apparatus and starts to interaction with the one of the proteins on its surface, like NS4A and NS4B, sorry, NS5B, and so these two proteins, so NS5A and NS5B, are used for the interaction, and then the RNA uses the cell mechanism for the transcribing the its proteins, and then will make a new capsid and then put it off, bought it off from the cell. And for the treatment, the 4 groups are effective for the treatment. And first group is protease inhibitors that include boceprevir, glisaptravir, razoprevir, and peritapravir, also cimaprevir and telaprevir and voxilaprevir. So these drugs are inhibiting the proteases NS3/4A, and that prevents the binding of the viral RNA with the Golgi apparatus, so they can transcribe the viral proteins. So, and the next group is Nssa inhibitors. So these include daclotazvir and elbasvir, ledipasvir, ombitazvir, biprentasvir, and dolutegravir. So these drugs are inhibiting the NS5A protein, and also the similar group is NS5B protein inhibitors, include dasabuvir, sofosbuvir, and sofosbuvir. So these drugs are inhibiting NS5B protein, also located in the Golgi apparatus. So despite the three steps of the entrance and also the uh, translation inhibitors, uh, also the interferons are effective for the treatment of the hepatitis, and especially on the chemically modified pegylated interferons alpha 2A and alpha 2A, alpha B, uh, usually in combination with ribavirin, that is an antiviral agent, and pegylated interferons are the interferons with that are chemically connected with the polyethylene glycol that increases the half-life in the blood, and that uh, prolongs the duration of action, because usually interferons, the human interferon alpha are not very stable and usually are destroyed of very fast. So, but they can help to uh, they can help to identify and uh, uh, to to prevent the action of the hepatitis C and B viruses, as well as other viruses, but mostly used in the treatment of the hepatitis. So, and the last group is herpes virus, and herpes virus is very common, and the drugs used for the treatment of the herpes virus are usually the analogs of the timidine, and because timidine kinase is an enzyme that will phosphorylate the uh, timidine, and that lead to the viral that is used for the viral DNA synthesis. And these drugs are chemically have a structure chemically relevant to the timidine, and that include acyclovir, ganciclovir, and penciclovir. And also they are acyclovir, they are analogs prodrugs that are acyclovir, ganciclovir, and famciclovir, that are converted to the corresponding drugs inside the body, and then they are recognized by the timidine kinase and used and they are phosphorylated and used for the synthesis of the uh, uh, nucleotides that are used further uh, for the building the viral DNA. And because of this, they don't have a timidine structure, so that's why that leads to the termination of the DNA synthesis, or the DNA will not be replicable, and either way something goes wrong with the DNA double strand, and then the virus cannot replicate anymore. So herpes virus is actually the uh, less viral and less contagious, and easily treated with any of these, uh, mostly acyclovir as the first of the drugs, but also any other drug, and usually is not a problem, while the HIV is non-treatable at the moment, and also the herpes, uh, hepatitis B and hepatitis C are also completely not treated, but require the lifetime treatment. For the influenza, usually does not require treatment, but actually it uh, can be uh, developed for the immunity can be developed against the influenza virus, and the vaccine is available for the prevention of the disease, but usually the immunity is capable of getting rid of the infection itself, provided it is not compromised, like in HIV patients or the patients receiving the immunosuppressive agents. So this is all for today, and thank you very much for your attention. And if you have any questions or comments, then please feel free to ask them below, and see you next week. Bye bye.